Good afternoon, everybody, and welcome to Cyrsiau Carlam Cymru. Welcome to the second biology session, led again this evening by Mrs. Paula Burt.
Thank you, everyone, and good evening to you. So in the second catch-up session in this series, I am going to talk about DNA again, but this time it's related to protein synthesis. So this is the second of the two major functions that I talked about with DNA. Just a couple of points before I start, in case you missed the session last time, just to remind you that the webinar is 40 minutes long. And you can ask questions in the chat bar. There's a chat bar available, and also in the chat bar there is a URL there so that you can access all of the materials and the recordings, and you can see I've put the link there at the bottom of the PowerPoint. Now, because of the nature of the webinar, I only have 40 minutes and I want to cover a lot of ground with you. So I will leave short pauses for you to attempt questions. But the idea is that actually, if there's anything that is unclear, anything that you just want to go back over, remember, when you get the materials, you can just go back over and work through everything at a pace that suits you at a time to suit you. So, OK, let's get going for today. So, coding for proteins. So what are the sort of questions we need to be asking ourselves today? So can you explain how the DNA triplet code works, is what I'm thinking. And are you able to explain how the proteins that are synthesized in, how proteins are synthesized in transcription and translation? And then also how messenger RNA, when it forms, can be modified, and how the proteins that are made can be modified. So those are our three areas of work today. So you may be used to seeing diagrams like this, and they basically show protein synthesis, okay? And you can see it's made of two processes, overarching transcription and translation. Now, the idea behind protein synthesis is that we have amino acids that are coded for by triplets of bases in the DNA. And then the DNA is transcribed to produce what are called codons in messenger RNA. And that is then translated out in the cytoplasm to produce a sequence of amino acids on ribosomes. So this is the idea about protein synthesis. And the idea is that a protein, the proteins are made in what's called a one gene, one polypeptide hypothesis. So for every gene, there's a polypeptide made. And this area of biology was talked about by Francis Crick, which you will recognise the name as in Watson and Crick. And he called it the central dogma of molecular biology. He gave a groundbreaking speech at UCL, University College London, in 1957, where he said, in a nutshell, I suppose, that the information in DNA is transferred to RNA and then to a protein. And so that's what we're going to focus on today. So if we have a little look at this diagram, what I'm going to do now is I'm going to take you through protein synthesis piece by piece. So first off, you can see when I'm moving my cursor here that actually this portion of DNA has begun to unwind. And actually the reason it unwinds is because the enzyme there, DNA helicase, is breaking the hydrogen bonds between the bases in the middle of the structure, as you'll remember. So the helix unwinds, and then exposed bases, nucleotide organic bases, are going to be, as I say, exposed to, in the nucleus there. Then what will happen is the RNA polymerase enzyme. So here we go, we've got a second enzyme, and here it is shown by this little circle. What happens is that links to this exposed part of the DNA, these exposed bases, and this part of the DNA is now called the template strand. And what it will do is it will move across the DNA molecule, this exposed part of the molecule here, and complementary nucleotides will be assembled one at a time according to the rules of complementary base pairing that I'll talk about in a little bit. And what will happen is bonds will be formed between the sugar phosphate backbone and messenger RNA will be formed, and this is called transcription. So that is the basic idea behind transcription. beyond the end of the gene. So by the time the RNA polymerase gets to this end over here, there will be a stop sequence. And that is a sequence that doesn't have any code for any amino acid. So what will happen is the RNA polymerase will leave that part there, and a molecule called PreMrna RNA is actually formed. Okay, now just as a link to later, that RNA, pre-RNA, messenger RNA, is called that because it contains exons and introns. Again, I'll come back to in a little bit. So the ribosome, so what we're doing now is that we're now moving this strand of messenger RNA out of the nucleus that you can see here, out through the nuclear pore, and we're now in the cytoplasm. And what happens is a ribosome will bind to a start codon. I mentioned in transcription about a stop codon. Well, there's also a start codon, and actually the ribosome will bind at that point to the messenger RNA. Now you can see the structure of the ribosome. It's got a large subunit that's got 2 binding sites for TransferRNA. And it's got a small subunit that provides a binding site for messenger RNA. And as I've said there in the box, the TransferRNA will bind to the ribosome, and there will be this codon-anticodon interaction. Again, it's all about complementary base pairing. So moving on from that, just a little something more about TransferRNA. So you can see at the top here what we've got is we've got some TransferRNA molecules, and each of these TransferRNA molecules is attached to an amino acid. Okay, now there are approximately 20 known amino acids, although that can vary depending what you read, between 20 and 27. So there is a specific TransferRNA molecule for a particular amino acid. So this TransferRNA molecule will have a very specific sequence of 3 bases at the bottom end of it there, called the anticodon, and that anticodon is related to the amino acid which is attached to the other end. So what happens is, as I've said, the TransferRNA molecules are able then, with their amino acid, to come in towards the ribosome. And as I've said, there are 2 attachment sites, and that's an important fact, that is, okay, because they sort of come in and the ribosome can hold just the 2 TransferRNAs at a time. So when the second TransferRNA molecule comes in with its amino acid attached, you can see there, the 2 amino acids are then close enough that a peptide bond can be formed between them at this ribosome. And you can also see, if we track back through, this has happened several times already, because a growing chain of a polypeptide is actually occurring there. So the ribosome carries on moving along the messenger RNA, 1 codon at a time, until the stop codon is reached, and then the polypeptide chain is able to break away. Now, what we formed at the moment is something called the primary sequence of a protein. Yes, because what we've got there is a sequence of amino acids. So we have a number of amino acids depending on the DNA that's unwound. We have a number of amino acids in a particular order, and that is what we call the primary sequence of a protein, just to link back to that topic for you. So this is translation. So transcription occurs in the nucleus, translation occurs out in the cytoplasm. So we've now formed our messenger RNA, as I've said, but it's in the form of something called PreMrna. And what happens is that we have to remove introns from our messenger RNA strand, because introns are part of DNA that actually are non-coding in terms of coding for proteins. So we've got a mixture there of exons, which actually have the code for proteins, and which will ultimately cause proteins to be assembled from the amino acids. But between those are the introns. I always think the sort of introns, in for intron, in for interfering with the primary structure of a protein. Okay, so what happens is the introns need to be removed. Now, I talk about pre-mRNA, but you find this happens in eukaryotic cells. So again, another little bit of a recap for you. Cells, plants, animals, fungi, protoctistans, these are all eukaryotic cells. And what happens is their RNA needs to be processed. This messenger RNA needs to be processed to get rid of the introns. So we've just got exons. And actually, you won't be surprised to hear that there's an enzyme that actually is doing that job. And this enzyme is called an endonuclease. And endo means within, and nuclease is referring to the nucleotides there. So an enzyme is able to get in there between those exons and actually remove the intron and then seal or splice the exons together there with a DNA ligase. Okay, so that is splicing. So the RNA that that we've got now is RNA that's fit for purpose. The pro cells, so of course this happens in eukaryotic cells, but in prokaryotic cells, the messenger RNA that's been transcribed is used directly. So there's no processing needed for pro cells, right? They generally don't have any non-coding sequences, so there isn't this process there, and that is something that is worth remembering. So what I've said there is basically what I've said already, in that the eukaryotic genes are usually discontinuous, and what I mean by that is the exon is the gene that codes, the intron is not. The exon is coding, the intron is not. So there's this discontinuous system of information along the DNA, whereas in prokaryotic cells, pro cells, the genes there are continuous, so the code can be read continuously from start to end. Now, once we've got the primary sequence of the protein, other things can happen. But before we get to that, I wanted just to go back and just say to you, do you know what? One of the most important ways to learn this topic is to do this for yourself, is to actually, never mind what it says in a textbook and the revision book you've got, try it out for yourself. So write your own DNA sequence, you make it up. You make up your DNA sequence with your A's, C's, T's and G's as you wish. Once you've got that double-stranded sequence, unwind the DNA and pick the coding strand. Once you've got the coding strand, then synthesize your messenger RNA alongside it using yourself as RNA polymerase. And then don't forget that the TransferRNA molecules have anticodons, which will come in and they will actually pair with your messenger RNA in this complementary fashion, and you will build your primary sequence of a protein. And you can see here the nature of the complementary base pairing. You can see the incoming TransferRNA with its very specific amino acid depending on its anticodon. And once the first of these TransferRNAs move away, then this second one can come in and a new polypeptide, the polypeptide grows as a new covalent bond is formed between the amino acids. And remember, in biology, we call that type of covalent bond between amino acids a peptide bond. Now, the TransferRNA is now outgoing, so it leaves that area there, but it needs to pick up more of the same amino acid, and it will do that as long as ATP is available. So again, another important reason for ATP being so significant in metabolism. Okay, so my tip is to do that for yourself. Now, there are 3 other key words that I haven't used yet, and you can see them below the diagram in black there. Initiation, so that's when the ribosome attaches to the start codon. The elongation, when the 2 amino acids are suddenly close enough then for that peptide bond to form between the amino acid and build the chain. And then the last part, termination, where the amino acids are added until we reach a stop codon, and then the ribosome can detach and it can move along the chain again, building another exactly the same polypeptide chain. Okay, so I just thought you needed a little bit more detail there of the complementary base pairing. Now, after we've got the primary sequence, so in my diagram here at the top here, what we've got here is the protein, having come off. So we've got the primary sequence of the protein, here it is, and all sorts of things can happen to it now. It can actually twist and fold to give different shapes. It can have molecules added to it. For instance, in this middle diagram here, we can see it's added sugars added to it. It's now a glycoprotein, and glycoproteins are really important in acting as recognition sites on cell surface membranes, for instance. The bottom diagram, you can see the phosphate has been added and that changes the shape. And as you and I know, when you change the shape of a protein, you change its structure. Just think about your basic idea there, induced fit, the active site of enzymes, and the shape there, that active site, is so specific to the substrate that it'll form a complex with. So, you know, translation is only the start of the journey really for the protein, because it'll be modified in order that it can fulfill all its functions. So we talked enzymes, but also don't forget we've got structural proteins in cell membranes, we've got hormones, we've got antibodies as well. So there's a whole host of functions the protein will then be able to fulfill. Now, there are other examples down here, and you don't need to know all the detail of these, but if you look at it, it'll give you some idea of the sorts of things that can happen to the protein. I have just mentioned one thing that is really important though, just here, and that is that the polypeptides can, once 4 polypeptides have come off, you can form a molecule of hemoglobin, because if you remember, hemoglobin is a quaternary protein. So this is a protein that actually is made of more than one polypeptide chain. So you can modify the polypeptides coming off, I'm saying, because then you can combine 4, and remember it's got a heme group at its heart as well, but you can make hemoglobin in this way. So polypeptides can be modified and we can make hemoglobin. So the genetic code then itself, well, we know it's a triplet code and we're used to seeing these sort of images, but what are the features? Those features there are key. Okay, so we say that the genetic code is linear, so that means it is read along in a line, one triplet after another triplet after another. We say it's a triplet code because there are 3 bases for every amino acid as part of the code. We say it's non-overlapping because as we go along the DNA, the code is read in 3, and then the next 3 are read, and then the next 3, and so on. So it's read as one triplet after another. We say the code is degenerate, and what that means is that not every, but quite a number of amino acids have more than one codon. So the first 2 might be AA, and then the third base might be C, or it might be G, but they will still code for the same amino acid. So that's what degenerate means. It means that very often you've got a bit of variety on that third of the triplet bases. The other feature is that it's unambiguous. So there, a codon will have one amino acid. There's only one amino acid for that codon, and it's probably a good idea to look at the words degenerate and unambiguous and make certain you're clear about the distinction there. And then the other thing is that it's universal. That code, that triplet code, will produce that amino acid, and that is universal. Now, different questions will show you the code in different ways, and they're all fairly straightforward once you've seen them a couple of times in how to work them out. So I'll just show you, for instance, the one on the left hand side to start with. So let's look at methionine, because I'll show it over on the right hand side as well. So this amino acid called methionine, MET, the first letter, so if we had the code AUG, for instance, we'd look at the first letter, right, that's A, so we'd look here, and then the second letter would be U, so we would look down from U, and so we would know now that it's one of the amino acids in this box here. But as our code, our third base is G, we'd look over here, the third letter, we'd go down, we'd see G, and so the amino acid will be met, or methionine. So that's one way of you being shown how to work out a code or how to work out an amino acid once you've been given the code. If you look on the right hand side, this like pinwheel shape, this looks a bit strange, but again, it's more or less based in the same way. So if we take methionine, which is AUG, what we've got here is A in the center. So we start in the center with A, and then we move out to U, and you can see there. And if we know the third base is G, then we know from this it has to be met, it has to be methionine. If we look at glycine, so glycine starts with a central G, you can see here. And then if we work outwards, it's another G, but you can see it could be all of these bases here as the third base would be the code for glycine. So you see some amino acids just have the one code and the others have more than one. So that's the nature of the code. Now, I also just want to point out that I've spoken about messenger RNA and transfer RNA, but I haven't actually gone into the detail of exactly what the different types are. So we can see here that we've got messenger, transfer and ribosomal, and they've all got these same features. Please remember that they're all single-stranded and they use uracil and not thymine. And you might be thinking, well, actually, isn't transfer RNA, doesn't that look a little bit double-stranded? And it's not. It's because there are hydrogen bonds. If you see in these images here, there are hydrogen bonds as the molecule is twisted, holding them together. Okay, so that's transfer RNA molecule, and you can see the anticodon here, here ACC, which has got the codon on messenger RNA UGG. Remembering again that there's no thymine in RNA, no matter which type of RNA it is, and that is very, very important for you to be remembering that in any questions. And actually, it's got the amino acid at the other end. Okay, so that is the structure of RNA, the three types. So I've actually talked for quite a bit now about all of these different processes occurring in the cell, and I thought it might be time for a quick little test, a quick little recap. And I'm just going to pause. I'll name each of these parts in place, and I've given you a starter for 10 there because I've given the game away with one of the answers. But have a little look. Where would you label messenger RNA? Where would you label transfer RNA? What about RNA polymerase? Is there something on there that you would say, yes, you know, I can recognise RNA polymerase from what I can see it doing? Nuclear pore, I think we've given the game away there for you. Ribosome, can you see where the ribosome is? And the other thing is, I've just talked about the three types of RNA, but the ribosomes are made of like a spherical ribosomal RNA, it's called. So that is full of ribosomal RNA. Where would the codon be? So where would the codon be sitting? Which strand? And where would you find the anticodon? And then can you see a polypeptide chain? And can you see a single amino acid? Now, some of these things appear in several different places, so that's no problem. Let's have a little look at where they're going to be. So here we go then. So messenger RNA, I've popped it there, but you can see, you know, you can label anywhere along messenger RNA there. Next one, transfer RNA. Well, we have a few transfer RNAs here. There's one here, but there are also others here that you might want to have labelled. RNA polymerase, so I've labelled the one here, but you can also see there's one there. So any of those are fine. Nuclear pore I've given you. Ribosome, you can see the large structure. I'm pointing to the smaller subunit at the bottom. And the ribosomal RNA, I've actually said, right, that's right in that ribosome there. A codon. Now, this diagram is a little bit small, so the codon, I'm trying to point to the bottom where I can see that CUG there, and the anticodon is definitely on the transfer RNA, so I'm sort of pointing to it there above, all right, attached to transfer RNA. Polypeptide chain, well, we can see that forming there, and a single amino acid we can see there. Okay, so I think that's just about all of them. Okay, so now that we've done our recap on transcription translation, let's see whether we can now look at some past paper questions. So Here we go. Name the enzyme involved in the transcription stage of protein synthesis. So I'll just pause. What's coming to mind? RNA polymerase. That's right. Now, you might have actually said their DNA helicase, and actually I would expect a DNA helicase question to maybe talk about unwinding the DNA. So I think probably RNA polymerase ought to be your go-to answer there. Then the diagram shows some molecules involved in protein synthesis. So we can see we've got 3 amino acids, X, Y, Z. We've got transfer RNA anticodons. We've got a messenger RNA strand and a DNA template. So let's look at these questions here. It says complete the diagram to show the bases on the template from which the messenger RNA was transcribed. So the bases on the template from which messenger RNA. Now, if you're going to transcribe messenger RNA from DNA, then you know there's complementary base pairing going on there. So if you look all along there, you can see C is complementary to G, G to C, and with DNA, remember, so T to A. Okay, so all the way along there, you've got the complementary base pairing. And then next it says, complete the diagram to show the bases forming the anticodons. So this is assuming you know that the anticodons are on the transfer RNA. So a little bit of knowledge there for you as well. So we know that the transfer RNA anticodons are complementary to messenger RNA. So here we go now. Have a little think, what is going to be complementary to GCA, sorry. And the answer is CGU, remembering again, no thymine on RNA. And we follow along to the next triplet and the next triplet of anticodons. Okay, so that is one question. So that is focusing very much on what's happening, you know, in transcription. Now this question focuses very much on what's happening in translation. It doesn't actually mention translation anywhere there. Okay, so, you know, let's have a little look at how we can get our marks here. So what we can see is our ribosome. So we've got our large and our small subunits. We've got our transfer RNA certainly here with our amino acid, and it's occupying one of the binding sites. But look what's happening, as I've said, to that second binding site. On this site here, we've got tetracycline, which it tells us is an antibiotic. And this is showing us how tetracycline binds to bacterial ribosomes. And explain, it says, how tetracycline stops protein synthesis in bacteria. So of course, if you stop protein synthesis in bacteria, bacteria can't survive. And so this is one way that you can treat a person with an antibiotic, because then the bacteria can't thrive, it can't grow. So don't forget the obvious here. They haven't mentioned the word anywhere. What's missing is the word translation, and then go into the detail. So the tetracycline is going to prevent translation. It's going to block the second binding site of CGC. Again, say exactly what you can see there. Think about the detail, and then you can't get the anticodon binding to the codon. No reaction. Okay, so you can't get the amino acid, the peptide bond can't form, and that is how obviously you can stop bacteria from dividing. Let's look at another question here. This one says the following figures show the messenger RNA transcribed from 2 short lengths of DNA. It says using the information following, complete the correct sequence of amino acids for each messenger RNA molecule. So we've got a template strand that they're telling us is normal. Then we've got a messenger RNA strand, an amino acid sequence, which we can work out from that using our table at the bottom. And then we've got another sequence. We've got a template, a messenger, but this time this is a mutated strand. So the one thing that you may notice if you've got time to sort of scan through there is that there are some differences between the normal template strand and the amino acid sequence in the mutation. And actually, if you look using the same method that I've told you about before, if you look at the table at the bottom there, you can see, so we've got our codes. It says codons found in messenger RNA, and that's another thing people do. They're not reading the questions, they're seeing we're looking at the codes on messenger RNA. So AUG, as I've said before, so the first base is A, so we're down here. The second base is U, so we're where A and U meet, sorry. And then the third base is G, and we've got it over there. So we know this first amino acid is methionine met. And you follow through with the others just like that, being very careful. And if you work out the second one, you can see the amino acid sequence hasn't changed. So it's then that's leading you into the next question, which is explain why the change of bases shown is described as a silent mutation. So this idea now they're drawing out of you that actually you can get some mutations, but some mutations may have no effect in the protein molecule produced. So there's no change in the amino acid assembled, no change in the order of them, so there's no change in the protein which has actually been made. So that is why it's a silent mutation. Right, on to another question now, and this one, it says in most eukaryotic genes, coding regions exons are interrupted by no non-coding regions called introns. And actually during transcription, it says the entire gene is copied into PreMrna. We've talked about that. That includes exons and introns. And during the process of RNA splicing, the introns are removed and exons are joined, as you can see here, to form in a continuous coding sequence, it says. Then we've got mature or functional messenger RNA. So all the things we've already discussed. Explain the process of transcription, including the roles of the enzymes involved. So we're talking transcription, we're talking about enzymes. So I'm hoping now, if I just pause, you can think of the two enzymes that I've told you about already. So think about those as I reveal the answer, DNA helicase. So we know the helicase is unzipping the DNA template. And of course, then we've got complementary base pairing occurring because it says explain transcription using the enzyme. So you want to explain how it is that those bases are then going to form, not forgetting that the RNA polymerase is our second enzyme because it does actually say including the roles of the enzymes. So it's not just one enzyme, it's leading you into mentioning two. So as we know, that'll join the nucleotides together to form that backbone of the messenger RNA molecule. Next question, explain why it's important that the RNA, which is finally translated, consists only of exons. So of course, remember I mentioned that the introns are non-coding, you know, the DNA that is not used to produce a protein. So exons are parts of the DNA which code for amino acids, so we need to think about that. Introns are parts which do not code. Okay, so that is why we need exons and not introns as well. And then finally in this question here, it says explain how a change in the sequence of bases in the DNA of the gene, that's a mutation, would affect the protein produced if it was in an intron. So now we know that the introns are non-coding parts, and it's asking us about if there was a change, if there was a mutation in the intron, how would that affect the protein made? So remembering what I said, the intron don't code for proteins, so introns, as they don't code, they will have no effect on the protein made. But let's think about what would happen with an exon, the coding parts. Yeah, now it's interesting that I've used the word might change. Okay, so if you have a mutation in an exon, it just depends. It might affect the primary structure, it may not. It just depends how much of a mutation, how many bases it's involved, whether bases have been repeated, or, you know, there's all sorts of things there. It doesn't necessarily, as we've seen in previous questions, cause the amino acid sequence to actually change in any way. Okay, one of the last of the short answer questions. I'm going to go through this fairly quickly so I can get on to the QER question. And I've said there how many different types of amino acids are used in protein synthesis. Well, it depends what you read. You know, I generally say 20. Why must there be 3? Well, if you think about it, if you only had, say, 2 base code, A and T, A and G, C and G, there's a limit to how many amino acids you could code for. No matter how much you try, you would not get code for 20 amino acids. So a 3 base code is more than enough, because you could get potentially 64 codes from that, and that is more than enough to code for our 20. Now, this question is a very similar question again, because you can see at the bottom we've got the same sort of table, and we've got our section of messenger RNA. My tip for questions like this, I mean, it's quite small on the screen ahead of us, so we can see the whole question. In the exam, it'll be a bigger piece of paper, or in your test. AUG, I would go through and I would actually section off every 3, because it's so easy when you've got a test to actually misread things. So I'd go through that as a little tip, and it says, using the information in the table, write out the sequence of amino acids in this section of messenger RNA. So we know the sequences are there, you know, so there we go. AUG is methionine, GUU is valine, and so on. You can put the rest in for yourself. Write the base sequences for the DNA. So now remember, we've got thymine, not uracil, but we've got thymine. So go through that and have a little check of that. And then the process occurring that gives rise to messenger RNA. Well, we've got transcription again, so I don't need to go over all of that. We've done that again. And then this one question at the end there talks about a mutation. And actually there's a difference here in these bases here compared to what's in the original section. OK, but this question is actually asking us about the point I made earlier. Explain why this mutation would have no effect on the protein that could be synthesized. Well, we've got no other information other than to think that 3rd base, because they differ in the 3rd base every time, this has got to be to do with a degenerate code, and actually the same amino acid will be synthesized, so the order is still the same. OK, so that one you might want to work over at your own pace again, but you know, I've been through it. So let's look at a typical QER question. So what I've done is I've just sort of thought of a QER question here. Describe how a nucleotide sequence on a DNA molecule results in the formation of a polypeptide. How polypeptides and pre-mRNA can be modified in eukaryotic cells. So I'm looking at transcription, I'm looking at modification of polypeptides, and I'm looking at what's happening in pMRNA. So just like I said last time, you'd find 3 parts to the question, and the 3 parts of the question sort of should be obvious. In this case, I'm hoping they're a bit obvious, in that the nucleotide sequences, I mean, that's going to be about transcription and translation. The second is going to be about how polypeptides are modified, and the third is going to be about pre-mRNA and the introns and exons, because it is mentioning eukaryotic. I mean, you could get a question about procells, and then you need to be very careful that, you know, there's no processing going on. So just like last time, my advice to you now is to think of all of the keywords that you can, put them in a stack, as I call it, a keyword stack. Put those words in a sensible order in sentences to try and craft your answer. But remember, marks in that top band, so 7, 8, 9 marks, you have got to articulate accurately. So, you know, you want to make certain you've got your keywords that you're explaining, you know, you're talking about them accurately. So you've got the words in a sentence, you've got them in the correct context, you've got them in a sequence that is sensible as well, so-called sequential reasoning. You don't want to miss any parts of the question. You don't want anything missed out. And if you're going to do a diagram, and this is fine, but remember, diagrams are going to cost time, so make certain it's not labelled. You see there, it's fully annotated, so you've said exactly what you're pointing at. And just like last time, what you may be thinking in terms of your keywords, base sequences, triplet codes, the code is for the primary sequence, the main ideas, transcription and translation. Don't forget our helicase, don't forget our RNA polymerase. Don't forget that we've got a coding strand, sometimes called a sense or a template strand. Don't forget in any answer to mention your complementary base pairing, but don't forget about RNA with uracil and DNA with thymine. Mention words like codons on the messenger RNA, anticodons on transfer RNA. Remember, transfer RNA will have its specific amino acids. Don't forget you've got the binding sites for the transfer RNA, that when they're close enough, the peptide bond can form. Also, remember that the transfer RNA will go off from the ribosome and it will be reactivated with a fresh amino acid using ATP. You may be asked about introns and exons, as I have in the question I've just given you there. And then you'll need to talk about endonucleases, actually cutting the introns and the ligases, splicing the exons together. And then the modification that I talked about, polypeptides, things being added, but also don't forget this question that you may have hemoglobin in it, and then you can relate it very, very directly to that. And I did promise you last week, so I'll just say one final thing, and that's about the die sheets, because last week I gave you a die sheet and I said, well, these are the sorts of questions you can use. And this week, these are the sorts of questions that actually we've been over. And I did say that, yes, you can have a template there to do your own. But actually what I've done this time is I've now provided you with those answers. So you can now use somebody else to help you with it and just randomly say 6 by 6, ask me the question that's 6 by 6, and whoever is helping you can actually then make certain you are answering with what's in that box there. Okay, so those are the die sheets, and I hope you found those really, really useful. So that's all I'm going to say about protein synthesis today. Next time I'm moving on to unit 1.6. I'm going to talk the first of 2 lessons about cell division. I'm going to talk about mitosis. So as I say, the next catch up is next Wednesday on the 17th. So don't forget to use the URL to access all of the resources. And this is not biology, this is a plea for you to look at other subjects as well. But thank you very much for watching. Good luck with your revision, and I will see you next week. Bye bye.